Let's just move right along to our feature presentation. So today, I'm delighted to introduce our featured speakers, Michael Parsons, PhD, senior researcher and consultant with the Center for Urban Ecological Solutions, and Luis Agurto, CEO of Pestec. Now, Michael's career spans over 2 decades in academia, including as an associate professor at the State University of New York, visiting research scholar at Fordham University, and research appointments at Columbia University and Hofstra University. He earned his PhD and completed postdoctoral work in Perth, Western Australia, where his entry into pest control began with an unexpected subject, the Western gray kangaroo. Upon returning to the United States, he applied the behavioral insights he learned there in Western Australia to urban rodent control in New York City, pioneering approaches that blend ecological understanding with practical intervention. Michael is a passionate advocate for bridging the gap between industry and academia and designs collaborative frameworks that enable pest management professionals to collect field data for researchers who in turn translate the findings into economically viable strategies for the industry. And Luis Agurto is a second-generation pest control operator and the CEO of the company Pestec. Pestec is an IPM firm that works with Bay Area municipalities, institutions, and businesses on sustainable pest management programs. Luis is also the structural IPM trainer for the Department of Pesticide Regulations Healthy Schools IPM workshops and provides hands-on training to school personnel. Luis holds a Bachelor of Science in Business Economics from the University of San Francisco and is an accredited certified entomologist by the Entomological Society of America and has over 27 years of experience in the practice of IPM. So today, both Michael and Luis will introduce our audience here to the complexities of rodent research and management, framing these challenges as a wicked problem that requires multifaceted solutions. Attendees will gain insights into real-world experiences managing and researching rodents in the field, and will learn about strategies to address rodent issues in the urban setting. So with that, I'll pass it over to Michael. So go ahead and share your slides and you can take it from here.
Okay. All right, thank you very much. And it's my pleasure to be with you on this early morning. For you guys, it's a couple of hours later for me in Houston. We're going to work into the lunch hour on my side. So I will start off with, we're going to immortalize love at first sight. So what you're looking at is a breeding pair of rodents from Brooklyn. And we're just going to immortalize this moment forever. And we're going to circle back to this a little later and what that means. Okay, so just moving forward. So the first part of this lesson, we're just going to move from pest control in Australia, where I began, where I got my start in pest control, and some of the lessons I learned there, difficult lessons that I brought with me to New York. And we're going to talk about the politics, because pest control isn't just about the science or the practice. You do have to navigate politics and relationships. And then most importantly for me as a researcher, the bane of my existence as a researcher is finding research specimens to study, because everybody wants to kill pests and not study them. And that leads to capacious gaps in our knowledge that really frustrates us. So my research looks a little something like this. My primary interest is olfaction. So I'm interested in the scents that make sense. So pheromones, charomones. We like to either increase or decrease trapping yields using scents or removing scents from traps. But we've not really done anything on a grand scale, right? We haven't created any alternatives or rodenticides. It's just moving the needle just one step forward. And something I do have 2 passions. One, like Jerry said, is to build bridges between researchers and the industry to learn to speak their language while teaching them our language, and then using the latest technology to do so. So we've microchipped rats. I think we were the first to do that in the wild. And we learned a lot of insights into rats because we got to know them as individuals and not just as numbers, okay? And so I hope to bring some of that to you. Okay, so my journey began in Australia, and we're talking about a continent the size of North America with less than 10% of the people. So, what kind of lessons from Australia in pest control can we learn? Well, first of all, in Australia, if you focus on the cities, the larger cities, the ones you're familiar with, Brisbane, Sydney, Canberra, Melbourne, you're moving along the coast, aren't you? All the way to Perth, where I lived for about a decade. And you haven't heard a lot about the Gibson Desert or the Great Sandy Desert or the Nullarbor Plain. That's because most Australians are 90% are urban. Guess where the pests are going to be? They're mostly urban too. The economy matters, and so the Australian economy is dominated by mining, energy, and agriculture. Okay, so pestilence. I'm going to introduce a mystery pest here. So I'll invoke suspension of the disbelief. Pretend you haven't heard my intro because I'm going to introduce a mystery pest. Pretend you don't know what that pest is. Okay, so the mining companies, they are required to blow holes in the ground, extract the ore, in this case, bauxite or aluminum, from the ground, and then they fill the ground. They are legislatively obligated to fill in those gaps and then to revegetate. Okay, so a big hole in the ground on the left should look more. Let's fire that back up. So the big hole on the left should look more like the right. It should be rehabilitated with biodiversity in mind, ideally with a native species. So so there is an organism that shapes this ecosystem, and mining companies are frustrated because they can't revegetate because of the selective palate of this organism. That is, if it prefers one food type, that plant doesn't grow. Okay. On the other hand, if it really devours a particular plant type, like a native plant that should be growing in abundance, it denudes that from the vegetation. So mining companies can't do what they're doing for the triple bottom line because of the pest industry and what it's doing. So all up, it costs about $350 million per annum to mining revegetation. And the pest also, it really detracts from the livestock because it competes with scarce water in this arid continent. So who is this pest species? All right, let's talk about the characteristics of any pest. All right, so many of them are nocturnal or crepuscular. They're more active at dawn or dusk. Many of them are generalists. They feed on a wide variety of foods, which makes them really difficult to exclude because they can eat quite a few of them. There is an old Aussie lingo saying that this animal will eat the shoes right off your feet. Okay. They flourish near human settlements. They follow the water, right? They have voracious appetites, and just like rodents in New York, population rebounds. Control efforts lead to population rebounds. Some of the political issues, poisoning. You have issue one, is it humane to the animal that's being poisoned? And number two, are other wildlife, secondary wildlife being poisoned in the process? Fertility control is gaining traction. Okay, so that's a plus in Australia. Exclusion, this really influences research and industry. Exclusion is really difficult. And you might say there's a 3-to-1 ratio between this pest and people. Okay. So who is this pest? Well, the European colonialists asked the Aboriginals, who is this pest? And the Aboriginal response was 3 words. And sorry, we might need to mute a microphone on there if we're able. There's someone, yeah. All right. So the European colonialists asked the question to the Aboriginals, what is this animal? And the Aboriginals' response was 3 words, and that was kangaroo. And that's how the kangaroo got its name, because the Aboriginals didn't know the animal that the colonialists were asking about. But this animal, there are about 90 million plus. You might be surprised to know that there could be 3 times as many kangaroos as people. And that's really hard to quantify because of the location of the people and the methods we have to count kangaroos on the ground. People have been to the Simpson Desert. They have used helicopters to take transects, and we have an idea of how many there might be in a given area, but it's really sparse, just like rats. So these are some compelling images, I hope are compelling to you. This describes pestilence in Australia. This is not to say rodents aren't a pest. They clearly are. But this is focusing purely just on kangaroos where I learned my lessons. Okay, these are roo guards on the upper left-hand side, animal traffic collisions, right? On the right, the golf course. On the lower right, I believe this was taken nearby Edith Cowan University. So, animals are on the campuses. They flourish near people, right? And there's all sorts of issues there. And then on the bottom left-hand side, I saved the best for last because I actually experienced as a graduate student someone from the Department of Defense ringing my office up and wanting to speak with me. And how could that be? And they wanted to know what I could do to help keep kangaroos off of their runways. And, at that point, I realized, pest control, what you guys are doing is seriously important stuff. And so don't downplay your role in the environment or to other people or to the animals themselves, because this is really important work. Unfortunately, I didn't have any solutions, but I was shocked at the question. Okay, so the impacts quickly, because we got to move to rats, right? So if kangaroos cost $350 million per annum, to some stakeholders like the mining company and agriculture, they're actually yielding $2 billion to the economy. Many of you will recognize the beautiful logo from Qantas Airlines. And it's because kangaroos are the national icon. You name your soccer and rugby teams after, the Socceroos. This is something you don't want to be seen to kill or control. You want to be advertising them. So lesson number one, competing interests depending on where you are on the spectrum. Okay. We also learned of, again, the poisoning. Monosodium fluoroacetate was the poison that was used in the early 2000s. It was really nasty stuff. And it was being dropped by helicopters and baits. It was killing non-target species, native wildlife, kangaroos, whatever consumed it. And, it became a political issue. Shooting, if you've seen the movie Crocodile Dundee, I don't want to overgeneralize, but, that kind of thing did go on, and it's not great optics, right, to have your national icon being shot in the outback. So it's a political issue. And so what we heard as researchers is we need research. Okay, we need humane alternatives to control the animals and effective mechanisms for control. So my task number one as a researcher was to find a research site, right? But how do you find a research site with wild pests? Who owns them, right? So take kangaroos, for instance. How do you keep kangaroos on a property that I have access to, right? Property is controlled by individuals. It's owned by individuals. I need to be there doing my research. What if the kangaroos move to an adjacent property, right? Well, Michael, you can build fences. Yes, I would agree with you, but also recognize that kangaroos can jump over fences. And so in this case, you just build taller fences. Why wouldn't that work? Well, I'll draw your attention to the right side of my screen here. And notice along the fence line, okay, there is a kangaroo crossing this fence. And I don't know if you can see it. I'll give you just a second to see if you can see the kangaroo crossing the fence. All right. And time's up. All right, right there it is. Because not unlike rats, kangaroos can collapse their skeletons and they can slide under a fence. Okay. So if they're not going over, they can go under. And so this was actually at my research site. And I call it the animal red-handed, okay? So research takes time, time equals money, and for most people, they say, come on, stop the research already. The pie in the sky. Lethality is easier than research. Just kill them. And so research became a highly emotional affair for me when I had to speak to homestead owners, okay? And I need to hear their perspective. And that was, we need livestock and dead bodies. All right. I'm no longer the good guy. I could be the bad guy because for me, keeping animals alive means for me to study means their livestock could be dying. So time equals money. Okay. So our solution, lesson number 3, and the most important one I learned was about building bridges, and that was aligning our research needs with the mining industry. We needed site access. So these companies, if you remember, they blow big holes in the ground and they have to revegetate with biodiversity in mind. There's some seriously good scientists at Alcoa and BHP Billiton, and they want to see a good job of their rehab. That's their one job, is to make sure rehab works. And so we partnered with them. They gave us access to their mine sites, trained us, gave us a four-wheel drive, and we gave them data, right? And so that's facilitated everything I've done since then. So did we solve the world's solution? No, I'm going to be honest with you. We quantified some of the damage, right? So that they could then take that to their stakeholders and apply for money, because we could quantify what was really going on. We learned which plant species had to be protected more. The kangaroo palate preferred some and not others. So we could actually plant, in some cases, sacrificials. However, if you plant sacrificials of any species in terms of pests, you might actually increase the pest population by planting sacrificials. So that was the give and take. Again, we're going to bring that right back into New York and eventually San Francisco. But for my work as a behavioral ecologist, we learn context within which predator scents might influence behavior. No, olfactory scents don't move animals, clear them from one paddock to the other. They can influence their behavior, and make them more vigilant, so that we can use a multimodal approach to sometimes attract or repel them. Look, animals are more vigilant in the open field than sheltered areas. It's common sense. So we didn't change the world, but we did something, and that was moving the needle forward. So lessons, just closing this down from Australia. Pest control is a social issue. It involves multiple stakeholders, bring them together. But most important, if you don't take anything from this presentation other than this, it's just that when we kill our research subjects, we accrue capacious gaps in our knowledge base. And so I want to show you those really embarrassing gaps of what we don't know in a couple of slides. First, moving forward to New York City, yeah, different landscape, right? But I had a tenure track position in the city. I was moving into academia, and it was time to find a new species because there aren't a lot of kangaroos in Midtown Manhattan. And so, but what they do have are a lot of rodents. So again, I partnered with the beautiful Ronald Sarno of Hofstra University and Michael Deutsch from Arrow Pest Control, and we just started building relationships between the industry and the research institutes, and we did some very basic fundamental science. So how are rats different than kangaroos? This is simple. All I did here was I copied the slide on the left hand and put it on the right hand and changed one word. That was it. Rats, nocturnal, generalists, flourish near humans, this voracious appetite, same control issues, population rebound, fertility control is gaining traction. You bet it is. We're going to talk about that. Does it work? Maybe, maybe not. Exclusion is a challenge, and most importantly, political. Okay. Now, I put this 4-to-1 ratio with a question mark. I don't count rats. I don't know anyone who can count rats, but it has been estimated that there are as many as 4 rats for every person. We don't know. But what I can say are the impacts about rats. Okay. Remember $350 million per annum in Australia for kangaroos? Well, we're talking 19 billion for rats. So this is a much more important industry in pest control. A lot of the expense is due to droppings, animal droppings and fouled food sources, but also public health issues and mental health issues. The majority of illnesses are not like the few leptospirosis happenings that get transmitted by the media. There's just a few of those every year. But the majority of rat vectored illnesses go unreported. And that's because when you go to a doctor with something like E. coli or salmonella, you're going there to be treated, not to be diagnosed. And so with the cause of the treatment. So if you have salmonella, it might be due to raw cabbage, right? Contaminated cabbage. It might be due to contaminated utensils, undercooked food, or it might be due to rats. We don't know, but we know it's a serious issue that's underreported. Food contaminations, but if you're familiar with the Baltimore study, get this. rat pestilence is more closely associated with depression in Baltimore than is violent crimes. I'll say that again. Rat depression is more closely, did I say rat? Okay. Human depression is more associated with rat pestilence than is violent crimes. And the reason the authors think that's the case is because you can do something. You can fight back from violent crimes. You can arm yourself or learn self-defense. At least it's something, right? But with rat pestilence, people feel completely unempowered, like there's another authority who takes responsibility for that. Not them. It's someone municipal or the pest control authorities. Okay. So infrastructure, briefly, fire marshal says 30% of all fires of unknown origin may be due to rats. Maybe, maybe not. We don't know. It's a guess. But you do know about infrastructure, wiring harnesses, and motor vehicles. Okay. So the answer is the same here and highlighted. We need research. We need humane alternatives. And I will add, we need to know the efficacy of these alternatives. We have a lot of alternative control tools. We just don't know if any of them work or to what extent they work. Okay. So my aims in New York were to collect data, fill a bunch of knowledge gaps, and for crying out loud, let's keep the subjects alive for just a little while, can't we? Okay. But finding sites was very, very difficult. And I just want to point out now, a couple of minutes into the slides, that we have a little toucan here on the right-hand side. And that little colorful toucan is just a reminder for me to remind you that in those of you who will be taking the assessment, that the toucan means that this is a test related slide. Okay. So when you see the toucan, yeah, that's a cue for you. Okay. So rats. They're small, nocturnal, difficult to identify. So a lot of people don't even know they have rodents. Or if they have rodents, they're not anxious to tell anyone, are they? Rodents are associated with fines, filth, disease, and oh yeah, depression. So let's hide them. Let's be ashamed of them. They're taboo, okay? The other problem with finding research sites, even if you say, Michael, I want to give you access to my research site because I really believe in what you guys are trying to do, there's still a liability we have to cover. And that is if people get sick with animals we're keeping alive, we could potentially be liable on some level. So this is a seriously, Lewis is going to describe to you what a wicked problem is. This is a wicked problem, okay? So if you remember the Australian homestead owner, you know, we need livestock and dead bodies. Does this look familiar? So in New York, from the time I was there until current, there's been this ongoing, very popular war on rats. Don't study the animals, kill them. And when we talk about rat math is the simplest equation you will ever learn. And I will tell you why this is complete nonsense and silliness. And it's wasting a lot of taxpayers' dollars. Okay. So the fourth lesson I learned in New York was, hey, there may be 10 million rats in the city, but no one's got any. You got to get used to rejection. So luckily, what we did have in New York was the media. Okay, so we had to partner with the media, and CBS, thank you, CBS. They helped us advertise that we were offering $1,000 to anyone that would help us study their rat infestations. Now, there were quite a few people who said, yeah, I've got this rat in my basement, or maybe it's a mouse, or I've not seen it in a while, but please come here and help me get rid of it. Yeah, there were a few of those, but there was also a waste recycling company that said, hey, we're interested in sustainable long-term rodent control. We don't care about killing the rodents now, as long as you can find an ongoing solution that can help us in the future. Wow. It was a needle in a haystack, thanks to CBS. Okay. So it's all about building bridges, isn't it? So I had to learn as a researcher to think like a business person. What is a triple bottom line? For instance, when I met Lewis, how can I speak his language? Okay, so I want to focus on business opportunities that my research can create. And hopefully you will think about your own businesses in this way. I want to help create control tools. I want to help evaluate control tools. And you might not have thought about this, but as a researcher, if I can help identify hotspots of rat infestations in your communities, you know how to target your rat control. You're no longer blind. And we have an app for that, which Lewis will describe to you. And lastly, pathogen control. Wouldn't you like to know if there were pathogens in high concentrations in particular areas in the community, things that could make people sick? I mean, I personally would go door to door knocking if I thought I could help someone out. So yeah, it's about making business allied to science and at the same time allied to the community. So that, to me, that is a triple bottom line. We're hitting on all cylinders. Just notice the toucan here. This is a test slide. Okay. Okay, and I need to be very, very clear. Why not laboratory rats? Okay, one word, no. I hear people asking me right now in the ether, okay, Michael, people are out there using rats to study the cardiovascular health of humans. Surely we can use laboratory rats to study wild rats. No. Laboratory animals are predominantly male juveniles. They're highly inbred. They're docile, tame, and they exhibit the same behaviors. They're bred to be clones of one another, right? Okay, wild animals, on the other hand, they're intelligible, they have very intelligent, variable genetics, they adapt to their environment, and they exhibit a range of behaviors. They think and act differently. So I like to use this analogy. Using lab rats to understand or estimate wild rat behavior is so close to using the domesticated Chihuahua from your living room as a way to estimate the behavior of wild wolves in Yellowstone National Park. That's not embellished. That's the truth. Okay. So that leads us to the state of the science. What do we know and what don't we know? Okay. And so there are a few embarrassing knowledge gaps. And as a researcher, humility it can go a long way. So humility tells a story. So I'm just going to tell you a story. If you were to ask me right now after our Q&A session and say, Michael, Mr. Rodentologist of 20 years in academia, real basic question, how long do rats live in the city? I don't know. Well, Michael, how many rats live in a colony? I don't know. And anyone who says they know is lying, okay? What about the ratio of juvenile to adults? Like, that's really important. Like, if we understand how many juveniles are per the adults, we can keep track of populations, right? Because if population control is working, there shouldn't be any juveniles. Come on, Michael, tell me you know. And the answer is no, I don't know, right? How do we influence and control their behavior? Okay, yeah, I've worked a little bit on that fourth point. I can make rats a little more vigilant through the manipulation of pheromones, and it works with some rats and not others. But I'm not controlling their behavior. Maybe something can be done with that information I'm learning. Maybe it can't. It takes time, okay? And here's our favorite moment of love that's been immortalized. This is our breeding pair again, okay? And so with that, what happens when you have a lack of information and knowledge gaps? Okay, and humble researchers telling you we don't know anything, or at least we don't know what we should know. And so headlines like this take over. And I don't know if you people, if you have heard of the cannibal rats that are just getting bigger. And by the way, they can attack our kids. You know, this is fairly popular during COVID. And without data, it's hard to respond to that. Luckily, Combs from Fordham University did such a study. They did something called morphometrics. They studied just the weight and length and size of rats and compared those skeletons to museum specimens. And you know what they found? Rodents were not getting any bigger over several hundred years that these specimens had been taken. So what happened? Possibly they opine that piloerection, the idea that fur stands on end, just like our hair stands on end when we're scared, that fur standing on end might make them look larger than normal. And if rodents are increasing in population, people might mistakenly think they're increasing in size because there's going to be more rodents seen by more people. And if there are rodents out in the daytime, for instance, taking chances to move around publicly populated spots, then it might very much, it would terrify the animals and it might scare the people. And I think that's part, Colmes opined that that's part of what's going on here. We need data. That's the only way we can tell you for sure. That's pretty important stuff, okay? So this leads to a bigger problem we have, and that's the non-validated control tools. And that means we have controls that we're implementing, but we don't know that they work. We're just spending money and putting them out there. And so that's the best example I know of anywhere for, putting the cart before the horse. Okay. And the best example of this, I think, of wasting taxpayers' dollars occurred in New York City. I don't want to pick on any mayor, but de Blasio administration in 2014, they decided to purchase $5 million worth of mint-scented garbage bags that were said to work in the laboratory. And but the issue was they can actually backfire, and they do backfire. Why? Because eventually when rats don't have an alternative, they literally learned that those mint scents are just strong scents that remind them there's food in the vicinity. So they learned, they associated very quickly. So what other control tools could we be wasting tax dollars on? Rodent contraceptives could be. I say could, because we don't know. If it works, it has an enormous, it could change the landscape. It could change the whole ballgame. But a lot of it's expensive and it's not been tested. Okay, so this is an image of our garbage bags. And so this is the next horizon. New York, all over the New York Times, if you've not seen it, and all the other major media outlets. They want to reduce the rat population using, instead of rodenticides, birth control. So this is the wish list for rats. Again, notice the little toucan on the right-hand side, test slide. We are trying to develop tests, or we call them assays. An assay is just a way to determine when a control tool is working or not. So we want to implement assays. Morphometrics, take the size and weight of animals. You're going to have a chance to help us out with that. Characterize stressed animals, trapped animals rather. Did you know that a stressed animal means something to rodent control? When I capture animals in Long Island, when I captured an animal that's stressed, it had this porphyrin, this waxy porphyrin over its eyes. It looked kind of greasy. The hair was matted. It starts in the eyes and it spreads all over the body. It's sebum, basically. And the more stressed the animal is, the more sebum it produces. Some of you who have cats recognize that. Rats are the same. We need to know if animals are stressed, because this tells us if our control is effective or not. Adult to juvenile proportions is so critical. And another thing we want to emphasize, carcasses. Pathogen monitoring is so important for our research, for public health research, and we think for your businesses, right? Tail snips, that's how we collect DNA, and DNA genotyping allows us to determine a lot of the population, where these animals have came from, where they're going, the size and range of the population, whether it's expanding or contracting. And most importantly, we want to be able to implement control tools that work. So this is probably the easiest rat math, the easiest equation you will ever learn. I purposely made it look complicated just so you can laugh when you see how easy it is. So the only context you need to know is, unlike people, rats are R-selected organisms. And an R-selected organism means they have a whole lot of offspring, high rate of fecundity, lots of pups, 12 pups at a time, but there's not much investment in each. So an R-selected organism, like an insect, you flood the environment with offspring, and you don't expect most of them to last, but a few will. That's an evolutionary stable strategy. Humans are K-selected. They have fewer offspring. They put more resources into a 9-month gestation, but they also expect them to last a little longer, right? So that's the difference between R-selected and K-selected. And so here is our equation on the left side. We've just basically, all this says is 2 breeding pairs of rats plus food, water, and harborage equals pups. Garbage in equals pups out. So a single breeding pair can yield, if the garbage is available, or food, water, and harborage, 12 pups every 8 weeks. So this, I'll demystify. Some of you have seen this diagram or one similar to that. If a breeding pair of rats give birth to 12 pups every 8 weeks, notice the toucan there, then after only 6 generations, right, 6 generations, you can have 112,000 rats. That's less than a year in 48 weeks. That assumes, right, that the rats aren't dying. There's unlimited food and no competition, right? So that's why we have to focus our pest control on the left side of that equation and not the right side. So we have to change the way we think about pest control. Look at the equation and never forget the simple equation. Okay, and I will just ask you a question just to circle down on this because it's so important. Okay, which side of the equation does rodenticides work? Okay, does it have anything to do with the breeding pair, food, water, or harborage? Or does it have something to do with the outcome? Okay, it's the outcome. It's the pups, the P at the end. What about trapping? It's the same thing. Okay, so how should rodent control focus? On the input or the output? And so that's really the simple message, and I think the take-home message is, as researchers, we want to primarily focus on this side, but also to be able to help validate tools that can influence the right side. So we want to have our cake and eat it too, okay? And ultimately, this silly war on rats, we have to stop declaring war on rats and by working on the right side of the equation, and instead change the behavior of people from the left side of the equation. And I have made snarky comments to the media before that we're declaring war on the wrong species. You know, I'm just trying to get people to think differently about this issue. But most importantly, we want to continue collecting data. So my summary to this point, look, we don't know enough about rats to control them, but we're trying. If we can find research sites to do so, we have to ally industry and academia. We have an idea to control rats. We know how to control rats. It's really simple. A simple equation can do it. But we also need research and data to know when control tools are working. And ideally, we can stop putting the cart before the horse and wasting taxpayers' dollars by implementing tools that we don't know if they work or not. And that will close down my introduction.
Thank you, Michael. Is my slide showing? Great. Thank you so much. So 27 years in this industry, I feel like I have paid attention. I have got CEU training. I have tried to learn, and I never heard about rats being an R-selected species until I started working with Michael. So, I mean, the knowledge gaps in academia are glaring and apparent and, you know, embarrassing, as Michael put it. My own knowledge gaps as a professional are even bigger and shocking to me. So Michael set the context for us of the wicked problem, but let's define it now. Set my timer. So what is a wicked problem? A wicked problem is one that resists simple solutions. Unlike tame problems, which can be fixed with straightforward answers, wicked problems are complex, multi-layered, interconnected with other social, ecological, and economic systems. They don't have a clear definition, and they don't have a single answer. And every attempt at a solution often creates new challenges. So classic examples would be climate change, poverty, and public health crises. So remember, there's a toucan on the screen. How do rats meet the definition of a wicked problem? Well, as Michael already set up, a simple answer is that there are conflicts of interest between the stakeholders that have to live with them, manage them, regulate them, and research them. And so now I'm going to go through some examples of these wicked problems with managing rats. Number one, dumping. Public use trash cans should theoretically cut down on litter. Where people can place their trash in a can, they won't throw it on the ground, right? Well, the problem is that the limited public use trash cans either become a magnet for residents to cut back on their own waste management expense by using this free service, or they become overfilled by legitimate public use to the point that they overflow onto the ground, and many times are not pest preventative by design, so rats and other wildlife are able to get into them and utilize those resources. So why don't we spend the money on pest preventative trash cans? Probably because they're expensive. Mismanaged waste areas. Waste areas in general are the primary driver of pests, rats in urban settings. Metal lids on dumpsters are less prone to fire, but heavier, and then therefore pose a risk to custodial staff in opening them each time they take out the trash. When the overfilled dumpster can't be closed, and when it's emptied by the waste management company, spillage is oftentimes left behind, feeding rats and other wildlife. So this is something that we're continually plagued with. We know that we have many initiatives right now to reduce our waste stream, which requires staging our trash, sorting that trash. And we are doing these activities or this operation in areas that were not designed for it. And many times that is directly creating a rodent problem for the buildings that we're managing. Another one is insufficient maintenance. Organizational boundaries between maintenance and custodial services may leave glaring gaps in facility operations. Additionally, when budgets are cut because of an economic downturn, oftentimes the first services to be downsized are the environmental services that keep properties healthy. These challenges, along with the tendency for working lunches or dispersed areas, mean food detritus is more common in more places inside buildings than ever before. So think about that. In schools, in offices, we're eating everywhere in those buildings. And increasingly, we have less labor, less support to be cleaning up after it. And so what do we have? A higher pressure for rodents inside of buildings. The next one, wildlife feeding. So feeding the birds and other wildlife is a natural joy for both children, the elderly, and anyone with a heart that doesn't know better. The famous bird feeder of Glen Park, pictured here, is an extreme version of this practice. And there have been more of these, from raccoon feeders in the parks to crow feeders at the beach, which are both prohibited by police code and park regulations, yet infrequently enforced. Backyard feeders across urban and suburban neighborhoods do as they intend. They feed the wildlife, including pest rodents that impact the neighborhood beyond the boundaries of entertainment, of those being entertained. Another one would be unfavorable design. So what I'm calling unfavorable design is any design choice made for aesthetics, cost, ease, or innovation that doesn't have long-term maintenance and pest prevention in mind. We could go on and on about this for a multi-hour presentation on this subject alone, and I could shame the well-intended architects, the facility managers, and the historical preservationists who refuse to kill their darlings, but we won't do that today. Suffice it to say that excluding rodents is oftentimes not pretty. Another one are the tree wells. Tree wells exist in the perfect place in between organizational boundaries. The trees and the tree wells themselves are managed by one municipal department. The lateral pipes that connect a private building to the municipal sewer is owned by the private property owner, and the municipal sewer is owned and operated by another municipal department. So whose rats are they? Everyone's? Who's responsible for them? Oftentimes, no one. And last but not least, the conflict of interest on most professional pest managers' minds is the studied and well-demonstrated repeated exposure to non-target wildlife from all anticoagulant rodenticides. What's the conflict here? Well, it's that the owners and managers of properties that seek to protect their properties and the public health, utilizing the chemical tools available to us, are in conflict with those that seek to protect the natural resources and who recognize an interconnected environmental health that these tools are damaging by their steady or without intervention increasing use. So I'm not going to spend a lot of time on a regulatory update. I think the professionals in this audience are well aware of the restrictions on anticoagulant rodenticides in California right now. I will suffice it to say that applications of these anticoagulant rodenticides are only allowed by exempted users or at exempted use sites. I will give you a quick update on the proposed regulations that we just learned about at the September 24th meeting that DPR held on anticoagulant rodenticide mitigation at a public workshop. Number one, they plan to further reduce the repeated exposure to non-target wildlife by making all anticoagulant rodenticides restricted materials. They want to limit where anticoagulants can be used for the protection of public health in agriculture and water. They want to limit the application to a maximum of 35 consecutive days with a maximum total number of days that you can use any anticoagulant at a site to 105 days. This would require, after the application of that rodenticide, to remove it after that period of time. They would also require training on sustainable pest management programs for rodents and a sustainable rodent management plan. This is one approach to tackling the wicked problem of managing rodents by removing the risk that these rodenticides pose. This requires enforcement. And as we talked about, there are many wicked problems that we're facing with that also require enforcement through that regime. So historically, I have seen that there has been a short-term approach to managing the wicked problem of rodent management, and that's what I call the crisis cycle. It's commonly recognized that pest management programs decay over time. For my opinion, what drives this is the same thing that drives entropy in all systems: dissipated energy. As buildings break down from the normal wear and tear of friction and routines of communication and operations are established to conserve precious human energy, nature takes over and makes use of the riches of our waste. The crisis cycle then unfolds, usually with the previous pest control company as the scapegoat, and with the new company pointing out all the pest sources previously ignored. With stakeholders and their attention at the table, new resources are allocated, the problem is at least temporarily resolved, and the cycle begins anew as it goes from a one-time capital expenditure back to operating budgets that are focused on saving money. The alternative that we proposed with this grant, and it's a mouthful, is the Effective Urban Rodent Ecology Knowledge Alliance, or Eureka, is a data-driven collaboration. Pest management service providers offer data exhaust in the form of surveys, access to research sites, and rat carcasses in exchange for deeper insight into the risk posed by rodents, data visualizations that help drive decision and policymaking, and research directly applicable to industry needs. Overall, the exchange is data for enhanced value. Eureka. This picture was taken at the South Coast Research Center in Irvine, where we had set up some sensors onto some existing bait stations, and we used some trail cameras to help validate that the sensors were actually capturing rodent activity. So now I'll go into more on this project and then in more detail. So the purpose of the project overall is to demonstrate alternatives to rodenticides and increase adoption of effective rodenticide-free strategies statewide. We're taking a three-pronged approach to this project. Number one, we're utilizing the IoT technology that's now available, and that's Internet of Things. So that's sensors and smart traps that are available to pest management professionals and municipalities alike. And we've been able to install 240 of these sensors over four large areas: 3 neighborhoods in San Francisco and the avocado orchard in Irvine that I had mentioned. This monitoring first approach, utilizing sensors that operate 24/7 with batteries that last up to 3 years and that limit the amount of boots on the ground time that it takes to do that monitoring, gives us a view of rat hotspots, peak hours of activity, and change over time as our source reduction recommendations and controls are implemented. The second prong of the project is the rodent log, which I'll go into more detail next. And the third prong of the project is to take the knowledge that we have acquired and bring practical tools to private properties to help them participate in area-wide rodent monitoring and management. We're calling that Adopt a Trap. So the rodent log is the tool that we've created and will be released hopefully in October, knock on wood, or perhaps a little bit later. And it's a professional citizen science data collection and sharing platform. It's a application that we made for Apple iPhones that you'll be able to download at the Apple App Store. And the first version is for use by professionals, both pest management professionals, those in environmental health, and in facility operations. Perhaps in the future, we'll have a new version for a broader audience. The goal is to turn the data that we otherwise would be exhaust that passes through our fingers and to structure that data in a way that we can turn it into real information that's useful for researchers like Dr. Parsons and all the other researchers that we're starting to interact with as this project has been unfolding. The first set of data that we're collecting is metadata that the phone collects automatically. That would be the date, time, a unique identifier of this person submitting the data, and the location of rodent observations. We're trying to make this as easy as possible for people to participate. So far, Pestec has pledged to use this application during this project, and we have some commitments from the Alameda County Vector and Mosquito organization and Orange County Vector Control as well. The only required data to submit would be 2 photos, one of the specimen itself and another one that's a context photo to tell us where did you find this rodent. The third one would be to identify if the rodent was caught in a trap or if you found it outside of a trap. The found state is an interesting thing for us to understand and to collect this information on, as the EPA, as it's undergoing its own rodenticide risk mitigation review, is considering making a requirement to do a follow-up after any anticoagulant rodenticide is used to see if you can collect poison carcasses and then prevent them from being scavenged by non-target organisms. So we want to test that. Is it possible? I know from practice that it is possible for Norway rats, but I'm a little skeptical that you'll actually be finding roof rats after a poisoning or even mice in some cases. We're also looking at the morphometric data that Michael Parsons is very interested in. So that's starting with the weight in grams of the specimen that's caught, then a length, which is from nose to tail, the size of the rodent, and then also a identification of the species of rodent that it is. Additionally, Michael had talked about, the physical condition of the rodent. That's something that I've noticed in my own practice, that some, rats can really look gross and have bald spots and scarring. And what I've learned is that that's an indication that those rats are under pressure. And that pressure could be environmental. Maybe there isn't a lot of food. Maybe we are restricting the resources that those rats need to thrive, or that the control efforts are otherwise making an impact. Other bonus data fields include site data. We want to understand where professionals are working, where are we doing our pest management efforts, and what are the signs that we're able to observe when we're carrying out those efforts. And those would include rubbings, latrines that are found, urine stains, droppings, and evidence of rodent feeding. We'll also be trying to collect as much conducive condition data as well, so we can start making a map of incidence of rodents in California and the key drivers. I'm telling you that in an urban space, I know that dumpsters are a primary driver, but that's anecdotal information. Working together, we can really validate that and then start thinking about how do we manage that in an appropriate way. thereby putting the horse in front of the cart. We also want to collect data on field methods. So what kind of traps are you using to collect these rodents? And how many traps did you set versus how many that you caught? And where were the traps located? Was it inside of the building, outside of the building, and specifically where? Hopefully, this information will be useful for other practitioners in the field to start adjusting their trapping in the future to yield higher results. And to gamify the use of this application and the bonus data that we're hoping users across the state will contribute to, we're planning a scoreboard to track the folks that we believe are making the biggest impact in managing rodents in the state. Although winning this recognition might be fun and perhaps something to share for their own professional purposes, that recognition is not helpful to rodentologists and public health researchers. It is fun, however, and we hope that it will be enticing for people to take that little bit of extra time, capture what would otherwise be data that's lost forever in a way that can be turned into information that's useful for researchers, policymakers, and the industry itself. So now I'll go into what we've learned so far. So far, we have set about creating our network of rodent monitoring or surveillance in these 4 large areas. This area is a residential space of small cottages or apartments. And we were able to create this heat map that you can see on the right here. The yellow dots are our monitor activity, and the one red dot is our high activity point. So we use the data to say, let's go look deeper at those locations. And what we found, was again, what we would assume, an open dumpster. So after conversion, having a conversation with this contact and talking about the data that we collected and these observations, we had a eureka moment. And the eureka came from the customer themselves in saying, this could really help us be precise with our communication. Instead of blanketing the community with, signage or notifications saying, please make sure you close the dumpster, we can use your data visualization, specifically pinpoint the area where this is actually driving the outcome that we don't want. And maybe with a little mix of public shame, we can start to influence the behavior of the people at that community in particular. So to avoid fatigue from signage everywhere that eventually people will ignore, we can be precise and use the data to tell the story. We've also learned that these heat maps can help us drive precision maintenance. It's not like this place doesn't have a landscaping team. They do. And that landscaping team is out working daily, prioritizing the things that they need to do in terms of risk management. So there's fire risk, there's tripping risks, there's things falling on vehicles. And in this case, if we're able to be very precise about a hotspot where the landscape is driving rodent activity, it's very simple for them to send the landscape team out there and trim that bush that creates a bridge onto the dumpster that lets rats in. It's such a simple idea, but by starting with monitoring first, then letting the data tell the story and reach the folks that we need to reach, we're able to be more precise and reduce the resource outlay and the fatigue that all these recommendations can otherwise create. Other interesting observations are that we're seeing rats using the rodent management stations that we're using for monitoring as restaurants. They take food from the dumpsters or take food from the environment around them and use them to feed inside of the dumpster of the rodent management stations, probably for protecting themselves. And so that's something that we're thinking about now. Do these rodent management stations alone, without a lure, have some monitoring capability that we can further reduce our resource outlay in filling them with non-toxic bait? And I'll talk more about that. And since we're working with scientists, we can't assume anything. So one of the first, challenges that we had as a researcher working with industry was, we can't count that the sensors actually mean anything because they haven't been validated by science. And so we're doing our best to do that now. And what we've seen so far is that our sensor data does correlate well with the consumption data that we're getting in those stations. So the more hits into the station or visits from a rat gets picked up by the sensor, and the more consumption of bait that we're seeing getting taken from that station. So we've also now deployed contraceptives in the assay that Michael was talking about. And I'll show you some video very quickly of observations in our bait station. So you see the bait station on the left here has a rat inside of it. That rat has been feeding on contraceptive bait and came out and perished. I'll show you one more time, so in case you missed it. We have been monitoring this location for about 6 months, only with non-toxic bait. We have not observed this happen before. We're now using the contraceptives for 2 to 3 weeks. We've seen the contraceptive get really demolished by the rats. They take it readily. And we now have observed 2 deaths from the product, or we suspect it's from the product. So our observation is contraceptive soft bait seems to be highly palatable to Norway rats, and we've seen 2 mortalities that we suspect are coming from it. Okay, we'll go back to Michael now.
I'm just going to
Okay.
interject with a quick time check. It's 10:40. I understand that both of you still have more slides to cover, correct?
Yes.
I can move pretty fast through this. Just looking at the research side of contraceptives. Yeah, I can hit the high points.
Okay, let's aim to wrap up by 11 so we have plenty of time for Q&A and discussion.
Too easy. Too easy. Okay. So as a researcher, you now understand why I'm the happiest guy in the room, or in the virtual room, as it were. Look, I'm thrilled. I have research sites now, thanks to Lewis and Eureka. And so how do I maximize what I have? I don't want to take this lightly after all the effort to get this, right? So the one thing I do, follow the money trail, validate control tools, and let's avoid wasting money that's not working. We all know about these olfactory products that, you plug them in, they excrete, they may or may not excrete a ultra high frequency noise that may or may not repel rodents. We've heard of these things. There's not a shred of evidence outside the laboratory that says they work. So we're interested in evaluating rodenticides. Which side of the equation do they work on, right? Some olfactory, auditory products, non-toxic baits. Let's find out if those baits are better than, for instance, some of the soft baits that are currently being used with rodenticides. But most important to me, I think, are contraceptives, because they really hold promise as a potential alternative to rodenticides. So again, this is all over the media, a lot of money.
Are you sharing your slides, Michael? I don't see them.
Okay, is it not showing? Okay, let's try that again. Thank you, Lewis.
It's okay.
Are we up now?
No.
No.
Okay, so we are hitting, let's try
There
that.
it goes. It's
third
going
time.
on.
Okay, so this is just a couple of images that I spoke about, the ultra high frequency plug-in, does it work or not, baits, and fertility control, right? So this has been all over the news. We know a lot of time and effort, resources is going into birth control. Does it work or not? What is our approach to answering this question in San Francisco, right? Okay, so briefly, our goals: fill a gap in literature. And hey, we know the laboratory trials worked with the contraceptive. Basically, the one we're starting with is cottonseed oil. You know about the cottonseed oil and the soft bait sausages. So the active ingredient is gossypol. It's found in human-grade cottonseed oil as well. However, it is extracted because that is the, quote, active ingredient. It's removed from the human consumed cottonseed oil, and it's left in there for the rodents. And it works in the mitochondria of the Leydig cells in the testes. So what that means is it damages sperm that's being produced, and it also disrupts the hormones. It can also affect females by producing, fewer pregnancies and smaller litters. However, first of all, we don't know what works outside the laboratory, and most importantly, it has to be continually consumed, okay? So among rats in the lab, there weren't any other foods to eat other than this. Okay, so we have our research questions. We have to know, do they consume this first of all, and will they consume it over other alternatives? What if they have a choice between a contraceptive and rolled oats and dog chow, right? Are they really going to consume the contraceptive? And how long will that take place? If rodents have to continually consume for it to be effective, it's just like human birth control. If you stop taking it, it's no longer going to be effective. They have to remain on this. So again, what does consumption look like over time? And most importantly, will it reduce the population size? And for how long, right? Because if you don't reduce population size, then it doesn't matter if they're consuming it. And then my question as a researcher is, what if just one breeding pair doesn't consume? For whatever reasons, will the offspring of that breeding pair, will some of those offspring develop resistance? That's the important question. That's how rodenticides, the first generation of rodenticides, obviously how they became resistant to those. So as far as our research, our number one obstacle was how, after we found the science, it was how do we identify population changes when we don't even know how many rats are there to start with. Again, I'm going to have this amount of humility here. I'm not going to pretend we're doing something we can't do. We have to estimate, and we have to do the best we can do. And in our case, we run something called cafeteria trials. That is, we put the contraceptive out in the bait stations that Lewis showed images of, and we have 4 alternating bait stations, and we go contraceptive, and then like a Nutella or dog chow, then contraceptive, and a Nutella. So we just compare the consumption in grams, how much is left over, right? And then how that affects changes over time. Is there a satiation or change in preference? Okay, and then when you replicate that over a number of sites, you can begin to make generalizations. Okay, so we're also importantly looking at incidents and behaviors. So 24-hour surveillance is so important for this research. We don't want to just measure the food that's eaten because we want to know who's eating it. And so we've marked out a 5-meter range around each station so that we can count the number of animals that are coming within that range and how the number changes over time. And importantly, this will help answer the question of, is one hungry animal eating most of the food, one alpha, or is it a bunch of other animals that are just picking at the food, or is it something in between? We need to know, because a lot of rodents need to be consuming contraceptives for it to work, right? And then probably the most important aspect of this, we're going to get a lot of information, but we also need to know the juveniles. And it's easy to tell how many juveniles are entering the field of view. And so we need to, after 8 weeks, right? If we think about our math equation, we need to see fewer juveniles in the treatment areas. But we need to compare those to the control areas to make sure that it is indeed the contraceptive that's changing. There may be something else going on that's causing fewer juveniles. So we don't want to say it's a contraceptive working when it's not. So as I just zoomed through here, this is what our research looks like in the urban environment: waste recycling plant, natural parks, and an ethnic community of food service establishments. If you come to me and say, Michael, I think I've got a research site, will you please study it? I'm saying, do you have a colony of 75 animals, ideally the same species? We don't want to compare Norways to roofies. We want to compare apples to apples. And there needs to be 24-hour surveillance. We need to be able to put our cameras in there and ideally minimize them from vandalism, okay? And we want to standardize this across sites so we can make the sites some very different sites, somewhat look similar in terms of the research design, okay? And so the design looks just like this. The image you've already seen, we're weighing the amount of food that's left over. And from that, we run cafeteria trials that tells us the preference over time. And then surveillance is helping us count the number of rats in this image. And that is going to, okay, and that is going to tell us, we're going to learn how many juveniles are in this image as well and compare that to the adults and how that changes over time. And so the design is going to look something like this. Don't get too hung up into it, but we have 4 microsites at every primary site. And each microsite has a single surveillance camera, 1, 2, 3, 4, from right to left, going from right to left. And then so these are the treatment points where we alternate the contraceptive with the control in the cafeteria trials. And then at the same point, we have a control point where there's going to be no contraceptive whatsoever. So if we see changes here in these 3 sites that we're not seeing in the 4th site, then we realize, hey, there's something different about this site versus the other 3. And then this is what it looks like when you look at all 4 sites. There's a minor discrepancy in the top, but ultimately you have 3 sites that are going to be the same. And then the 4th site is going to mimic the 4th microsite, which means it's just controls. And if something is going on in the 3 overarching sites that isn't happening at the 4th site, that again gives us some anecdote that something population-wide is going on, okay? So 3 levels of control, the amount of food consumed, ratios spotted, ratio to juvenile relation ratio, and we're also collecting DNA samples, genotyping, just to characterize the populations. And we hope to produce the first ethograms. In science, for our science geeks out there, you're probably used to ethograms that categorize behaviors and explain them and produce diagrams, because we don't even really know what rats do in the wild, right? So we want to be able to document this for the first time. And so ultimately, what will we learn? Well, hey, it's really hard to prove that contraceptives are working, but we will know if it's definitely not working, right? We're going to know if those populations stay the same and there's no difference between either level of treatment or control. We know it's not working. And so for me, it's the addiction. It's a research addiction that drives me and many like me. It's like I want to. Information is too important. It's complicated, but it's too important not to make a good attempt at finding. So, and most importantly, I think, if this works, we're going to use these same trials to evaluate other control tools, okay? Not just contraceptives. So we will use it on olfactory tools, auditory tools, baits, you name it. And ultimately, hopefully justify additional trials. So with that, Lewis, I will just pop this back in your direction.
Thank you, Michael. Okay, so next steps. Michael shared with you the contraceptive trials that we're doing. In the project itself, our next steps would be to do a necropsy of the suspected contraceptive soft bait mortalities. So that was something that we didn't necessarily expect, but are very excited to find out what killed these rats, and if it does trace back to those contraceptives, and then to speculate what that might mean on the contraceptives and how they'll work in the field as well. The other thing that we've already started is the Adopt a Trap program. Again, the idea with this is to take the same tools that we're using for monitoring and managing rodents in public spaces to the private properties where we suspect these rodents might be originating from. Before we had launched this project, we had done some other work in some communities in San Francisco, reaching out to residents and private business owners, asking them, number one, if they had a problem with rats. If they did they report that problem? And if not, why didn't they report the problem? And overwhelmingly, yes, people had a problem with rodents. They didn't report the problem, and the reason they didn't report it was that they were worried about putting their neighbors in any kind of problem with authorities. So the idea with this program instead is to give people a tool that's very practical that they can use as they would any other rodent tool, but that engages with us with the data collection side so that we can bring our data visualizations into this and then take the resources where they're most needed. We have canvassed different neighborhoods, reaching different property owners. We have a multi-unit building that's working with us, a grocery store, and a office building, and some industrial sites so far. And we'll be working in the community of residential apartments as well, utilizing sensors in the crawl spaces or attic spaces, using Internet of Things smart traps outdoors or inside those same spaces to monitor rodents, catch rodents, and then indicate when that trap needs to be resurfaced. So the idea overall is start with monitoring, guide resources where they're needed, and service traps when you need to, not by routine, where many resources go that could be otherwise spent doing inspection or communicating with your stakeholders. Another next step that we have is to start testing area-wide monitoring with no lure or with a decoy lure. So this slide that I'm showing on the screen that shows that an 83 to 104% increase in device success with the use of information decoys. What that means is it's a food lure that's inside of a salt shaker or inaccessible container where rodents encounter it before entering a station and then want to get into that food, and then we'll take them inside the station for that resource. So we want to take this trial that was and shared with us by Peter Banks in Australia and put it to use here in San Francisco where we can, to see where we can further reduce the labor input for monitoring. And then we'll also be implementing our non-rodenticide controls. So now that we have a baseline of data for activity in the areas using our sensors, we'll start to implement a variety of alternatives to rodenticides or non-rodenticide controls, including the contraceptives, as I already showed you, but also gases and other things. Here's a quick video of the use of gases. This is the CO2 treatment. It takes about 3 minutes per burrow to, following the label, to do a treatment with these gases. So we have a technician demonstrating that there. While that's running, we're also using a smoke cartridge to treat another burrow on the property. So there's pros and cons for both of these techniques in terms of price, the ease of using the equipment, fire hazard, and the time that it takes to do the work. I just wanted to share with you that quick example. Additionally, we'll be carrying out different trapping programs. We'll be doing some pulse trapping. So that's where we go at nighttime or over the course of the weekend and set lots of traps to cull a population and repeat that for a period of time. One of our sites, we've been doing that for several years and really achieving our goals for rodent control there with that. And we'll be keeping in mind the humane treatment of animals. Remember the toucan. Humane treatment means the minimization of suffering. Recently, the San Francisco Commission of Animal Control and Welfare submitted a letter recommending banning glue boards in San Francisco, I think on San Francisco city-owned property, for the use of glue boards because of the inhumane treatment of rodents. Our focus and attention will be on humane measures to manage rodents, and really wanting to emphasize that there will always be some management of rodents that will include lethal control, but I think that we can, as much as possible, make it as humane as possible so that all our stakeholders, so we can have less conflict with our stakeholders. So our conclusion, wicked problems are people problems. Honest and equitable collaboration is the key to solving them. And that's what we're demonstrating with this relationship, not only with research and industry, but also with all the stakeholders that we're working with here in San Francisco and as well as at Irvine. Number 2, data visualizations enable precision communication and maintenance. Precision saves resources and avoids fatigue. Number 3, soft bait contraceptives seem to be highly palatable and potentially lethal. More research is needed. And number 4, crisis avoidance requires adapting to change now. You all know this, but we should be proactive and not reactive, and that's ultimately the call to action. Please join us in this collaboration. The rodent log will be available for you soon. If you have research sites that you'd like to engage with Dr. Parsons, you can reach us at our partners at eurekaalliance.org email. You can follow the QR code. We'd love to work with you. We'd love to see the work that you're doing, capture that, visualize it, and use that data to better inform decisions and policymaking in the future. Thank you.